So in chapter 11, we looked at different ways that we can inhibit or kill microorganisms from growing, generally on the surface of things, whether on tabletops, on counters, whatever. It's inhibiting microbial growth on the surface. Chapter 12 is all about inhibiting microbial growth, but now in the body. So it's the different drugs that can be taken to inhibit microbes from growing. Now, the chapter is going to talk about different kinds of drugs. Where did they come from? We're going to talk about what selective toxicity means, how they work against these microbes. Again, they're going to inhibit or kill the microbes, but how? We're going to look at the toxicity or side effects that can happen when taking an antimicrobial drug, the whole issue with drug resistance, how we choose an appropriate drug. Because if you're going to administer some type of antimicrobial drug, you want to make sure it's the right one and in the correct dosage. So starting off with just, what is antimicrobial chemotherapy? Again, chemotherapy just means using chemicals to treat something. And in this instance, we're going to use some type of antimicrobial drug to treat infected individuals against some type of pathogen that got in the body. Now, some of these drugs are produced naturally. They're found out there in the environment, and they have different components in them that inhibit microbial growth. And some of these drugs are man-made. They're synthetic. They're made in a lab to stop microbes from growing. Now, the ideal antimicrobial drug. And again, we ask a lot of our drugs. One, they're selectively toxic to the microbe, which means they are specific for the microbe we want to kill and non-toxic to us. They're microbicidal, which means they're there to kill rather than stop growth, the microbistatic. They remain potent long enough to make sure they kill all the microbes, but they're not broken down and excreted prematurely. So we want them in there long enough to kill everything, and then pretty much only that length. They're not subject to antimicrobial resistance. They assist or complement other activities of our immune system. They remain active even when they're diluted in our body fluids and tissues. They're easily administered, they're cheap, and they don't cause us any harm. They're not going to cause us any type of allergic reaction or any other issue. So we ask a lot of our antimicrobial drugs. Now again, they're not all going to fit all of those different components, but that would be our ideal antimicrobial drug. Now, an antibiotic is generally, these are naturally produced or naturally found substances that are antimicrobial, so they inhibit microbes from growing, and they come from different bacteria and fungi. So you wouldn't think that fungi, which is a microbe or can be, and bacteria, which is a microbe and can be, they can actually produce antimicrobial agents. It works for them because depending on where they end up, there's only so much food, there's only so much space. And so a lot of organisms have figured out how to inhibit other microbes from growing so that they've got full access to those resources. This is showing a couple examples, some of the types of microbes that give us different types of or the different microbes that give us different types of drugs. So Penicillium is a type of mold, and different bacteria give us different types of antibacterial drugs. Now you might recognize Penicillium, but you might not recognize some of the actual organisms out there that make the drugs, but you might recognize some of the drug names. And your drugs, those antimicrobial drugs actually came from some other type of bacteria or some other type of fungus. Now, some terms when we talk about antimicrobial drugs, and the ones that have the little stars are defined on the next slide. But again, an antibiotic, these are naturally found substances, whether in a bacteria, fungus, or somewhere out there in the wild. Semi-synthetic is pretty much an antibiotic that we tweak in a lab, so we modify it. Maybe it's to make it last longer, more shelf-stable. A synthetic drug is an entirely man-made drug. Now, a narrow spectrum of action means that the drugs worked on just a small number of microbes. Sometimes even they're specific to one particular microbe. Broad spectrum antimicrobials work against a large number of microbes. But everyone's like, wow, well, that sounds great. That they can in itself have poor side effects. Now, this is those three that were defined from that previous table. A prophylaxis, this is something that can be given to prevent an infection. This can happen if someone's immunocompromised and they know they're going into a situation that they might be exposed to a particular organism. They would give you a drug beforehand. This happens for those that have weak immune systems before they go to the dentist. Because when you go in for a dental visit and they are poking in those gums, and if they start bleeding, that's an open wound for microorganisms to get in. And if you're immunocompromised, you won't have the immune system to fight off those now invading microbes. So they're given a prophylactics. They're given some type of drug to prevent an infection. Chemotherapy, again, is using these drugs to treat a disease, and an antibiotic just means anything that inhibits or stops growth of something. Now, again, we do want our antimicrobials to be selectively toxic. We, again, want them to kill or inhibit the microbe without damaging us. Of course, we don't want to be harmed by taking these drugs. We want only the pathogen to die. Now, that can get trickier as there are some pathogens that start to have very similar processes that we do. So then it's hard to target destruction of one thing without having side effects. This can happen with a lot of our eukaryotic microbes. Now, how a drug works. There's 5 major ways that drugs kill. They do not all kill the same. So the first group, and we're going to talk about, my next slides are going to talk about how they work, but some drugs inhibit cell wall synthesis, some inhibit cell membrane structure or function, some inhibit the making of DNA or RNA, some drugs stop protein synthesis, and some drugs stop some type of metabolic pathway. So there's different drugs to stop different organisms. So this is to show kind of those 5 basic groups. There are drugs that stop cell walls, cell membrane, DNA, protein synthesis, and metabolic pathways. And then it also gives some examples of some of the drugs found in each of these groups. Cell wall inhibitors, and I'm like, is usually the most common way we try to inhibit bacteria from growing. So you may recognize some of the drug names. You might recognize some of the drug names in the other groups as well. Again, it's just how they work and how they kill those different microbes. Now again, a narrow spectrum drug means it's effective only against a small range of microbes. Some example down here, polymyxin only works against some gram-negative bacteria. It's not going to be effective against any gram-positive bacteria, any of these unusual bacteria. It's not going to work against any eukaryotic cell. It's very specific. It only works against a small range of microbes. The opposite, a broad spectrum drug, has a greater range of activity. Erythromycin works against all gram-positive bacteria, most gram-negative bacteria, and even some of these kind of unique bacteria. I'm like, it's a broad spectrum drug. Now, the drugs that inhibit cell wall synthesis, again, some of the most common drugs that are given out there. Now, the cell wall of bacteria is made up of the substance peptidoglycan. It's made up of all the NAMs and NAGs. And in this picture, it would be all of the peach-colored and all of the orange-colored little hexagons on here. That's your NAMs and NAGs. Now, in a normal cell wall in a bacteria, they have all of these little cross-links to attach them. Again, a cell, any kind of wall, if you're going to build a wall with bricks, you're going to want to put mortar between each of them, because if you remove some of the mortar, as some of these green links are gone, if you remove some of the mortar, it's not a very strong wall anymore. And what can happen is that wall will now burst with some type of osmotic pressure. And I'm like, that ultimately will kill the cell. So if you can inhibit the making of peptidoglycan correctly, that it's not made correctly, you can stop cell walls. And because our cells don't have peptidoglycan, our cells don't even have cell walls, this is very specific for bacteria, which means there should be limited side effects. Just showing another picture of all those NAMs and NAGs. They're attached to each other sideways, top down, front, back. But again, if you take one of those drugs, such as penicillin, it removes some of those links. It makes it a weaker wall, and those bacteria will die quicker. Another mechanism of action is drugs that are going to somehow disrupt the cell membrane. Again, a cell membrane's job is to allow things in that needs in, allow things out that needs out. That's your passive and active transport. But if you somehow disrupt that, it ultimately will kill the cell. This is just showing an example. A lot of times these drugs are going to insert pores or channels where there shouldn't be any. That causes leakage. Things are going to get in that shouldn't, things are going to get out that shouldn't, ultimately kills the cell. Showing another example, polymyxin, it's linking to various types of, carbohydrates, and again, it's causing the disruption of the cell membrane. Things are going to get in that shouldn't, things are going to get out that shouldn't, and it disrupts the whole process. A third mechanism of action is anything that affects nucleic acid synthesis. That's the NA of DNA or RNA. Various types of drugs, and they can work differently. One common way that they can work, though, is that these different drugs can insert what are called analogs of your purines and your pyrimidines, your adenine, cytosine, guanine, thymine. Analogs, these are lookalikes. So they look like adenine, they look like thymine. And so these are showing some of the examples of some of the structures of these lookalike bases, and how they can have similar enough structure that they get brought in instead of the correct base, which means different viruses, these are usually used for viruses, different viruses, when they're trying to make new DNA or RNA, they bring in the correct base. It's similar enough so it lines up, but it's not the actual adenine, thymine, cytosine, or guanine, which means you've now made mutations, which ultimately will kill the virus. Another group of drug or mechanism of action are the drugs that stop protein synthesis. Now, they're going to target the ribosome, and their job is to somehow stop translation. So they target the ribosome and they stop translation, because you've got those two steps of transcription, making messenger RNA, and then translation, taking that messenger RNA and making protein. Well, the ribosome is what reads that messenger RNA to make protein. And again, proteins are used in structural material. Proteins are used as enzymes. If you cannot make proteins, you will not survive. Now, they can target ribosomes and translation several different ways, shown by these different pictures. This one is going to stop the correct amino acids from getting brought in. This one is going to stop the transfer RNA from being able to dock, which means it's not going to be able to bring in and attach the correct amino acid to it. This particular drug is going to stop amino acids from getting linked together, which means even though you bring in the correct amino acids, if you can't attach them, you're not going to make a protein. This one stops the messenger RNA from getting brought in. Another drug can stop the ribosome from even attaching to the messenger RNA to get read, or you can have drugs that stop part of the ribosome from coming in and attaching and reading the messenger RNA. Either way, if your ribosome can't read that messenger RNA correctly, you're not going to have translation take place. And the last way that drugs can affect, or the last mechanism of action, are drugs that affect different metabolic pathways. Again, there are lots of steps that have to take place to make a correct product. And if you have a drug that stops anywhere in that process, you're going to affect that metabolic pathway, and the correct product is not going to get made. Now, one way is you could do this by competitive inhibition, that the drug is going to compete with some normal substrate that an enzyme needs, which means an enzyme that's needed somewhere in the process can't get made, and it can't do its job, so it's not going to make the intermediate product so that the final product can't get made. Now, a lot of times what we do to try to increase the chance of interrupting the metabolic pathway is we use what's known as the synergistic effect. It's using a combination of antibiotics, because the combination is greater are greater than the sum of each individual. As they all work together, they can stop different metabolic pathways in multiple different places. Now, altogether, there are around 280 different antimicrobial drugs that are all grouped into 24 different drug families. And a lot of the ways that they're grouped is how they work. There's different drugs that act on the cell walls, that work on the cell membrane, that work against the DNA or RNA, that interfere with protein synthesis, and that block the metabolic pathway. The problem is, the more antibiotics we take, the more that these organisms are becoming resistant to these different drugs. So the more we take, the more they're not going to work anymore. So drug resistance is just a response where the organisms can either tolerate the dosage that's given, or they've completely figured out a way to avoid the drug. The drug's no longer going to be effective at all anymore. Now, how do they become resistant? Well, it can happen. Spontaneous mutations. I mean, generally mutations are bad, but occasionally a mutation may happen that's a good mutation. And these different organisms are now resistant to a different or some specific type of drug. That's rare because, again, generally mutations are bad, but it can happen. More commonly is that these different organisms are going to acquire new genes, new DNA, sometimes from another species, and all of a sudden they now have DNA that makes them resistant to some type of antimicrobial drug. Now, generally how they get these new snippets of DNA, these new gene, is they're using bacterial conjugation. It's the number one way. It's these little plasmids, these little snippets of DNA found in a bacteria cell that don't code for normal bacterial growth, but they are still DNA and can still have information to make organisms resistant. And it can happen that these little snippets of DNA, this little orange snippet, can go from one bacteria to another through bacterial conjugation. Sometimes they can go from one bacteria to another using a virus. And sometimes, and I'm like, they can go from one bacteria to another through transformation, meaning that DNA gets released and you can have DNA going from one cell to another. Either way, when bacteria talk and when DNA is getting moved around, it's never good for us. It could be good for them, but never good for us. Now, how they become resistant. To say they're resistant, it's like, oh, the drug doesn't work. Well, there's different ways that now the drug doesn't work, how they've actually developed this resistance. One is they can inactivate the drug, is that they can take a drug like penicillium, and by using an enzyme, they can actually break apart the active part of penicillin, and now penicillin doesn't work anymore. So they might make some inactivating substance to inactivate penicillin. Some it's decreased permeability. A lot of times the drug has to get inside the bacteria for it to actually cause damage. Well, if the bacteria or the microbe can prevent the drug from getting in, it can't do its job. So it could be an example. This is the normal receptor that that drug sees, recognizes, attaches, and then gets inside the cell. Well, if the microbe is like, well, I'll just change that normal receptor shape. Drug can't recognize it, can't attach, can't get in, and the organism lives. It could be activation of a drug pump that once that drug gets inside that microbe cell, the microbe's like, oh, well, that's not good. We should somehow pump that right back out. And so it will now activate a pump, and it's just going to pump the drug right back out. Drug can't kill. It could change the drug binding site. And so where the binding site on a ribosome, and I'm like, maybe that drug was supposed to work against ribosomes in translation, but now the binding site is altered so the drug can't bind. So if it was normally supposed to bind right here to stop protein synthesis from happening, well, it's now been altered where that drug can't bind there, and protein synthesis is going to take place like normal. And another is they can alter their metabolic activity. So we have drugs that try to inhibit different types of pathways, that if A needs to become B, then needs to become C, then needs to become D to eventually make a product. If we can inhibit B from becoming C, that organism can't make their product. Well, that's great. However, some organisms have figured out a way around it, literally around it. Well, it can't make normal C, it makes C1, and that makes D1, and then it eventually makes its product, and so it still survives. So organisms are literally figuring a way around it. Now, what can happen is the more you expose an organism, to a drug, the more opportunity you're giving them to figure out ways around these drugs. So it's called natural selection, that all the time in any large group of organisms, there's genetic diversity. And all it takes is one organism to have the correct gene to inhibit or stop that drug, that organism's going to survive. So this is showing a whole bunch of different organisms on there. They can be the same organism, but genetic diversity-wise, these two little guys have some type of DNA that allows them to be resistant to the drug that's given. Once the drug is given, all the little non-drug resistant cells are all going to die, but the ones that are going to survive are these guys. They have the DNA. They have some type of genetic diversity that allows them to be resistant to that drug, and they're going to do what they do best. They're going to reproduce. And so with continued exposure to the drug, generally you're going to have natural selection. The strongest or the smartest are going to be the ones that survive, and now you've got a whole bunch of new bacteria that are all resistant to that drug. Now, to try to stop this from happening, the number one way, well, I don't even want to say the number one way, a top way, is drug usage. That only the correct drug should be prescribed. That we're not prescribing drugs that don't have any effect on the organism. Because again, the more you expose organisms to different types of drugs, the more opportunity you give them for them to figure out ways around the drugs. The patient should take the correct dosage by the correct or the best route for the appropriate period. Again, if we go back to this picture, those are drug-resistant cells. But maybe if you actually took the full dosage, the correct dosage, and you took it for the entire period of time, that by day 10 out of day 14 of your drug prescription, by day 10, those two guys finally died. Because right away, the ones that are going to die the soonest are going to be the weakest one. Because when you first start taking antibiotics, if you're given a 14-day prescription, you're usually feeling better by day 2 or 3. And so a lot of individuals stop taking the drug, thinking, well, I don't want to keep taking it. The problem is, by day 2 or 3, these guys haven't died yet. It takes 10 days before you've given them enough drug to finally kill them. So if you stop taking the drug at day 2 or 3, these guys are still going to survive, and yeah, they're going to reproduce, and now you have bacteria that are stronger, more resistant to drugs. So you got to make sure the correct drug is taken, and it's taken appropriately, and that might even mean taking a combination of drugs. Again, the whole goal is to kill that pathogen, and sometimes you need to take a combination of drugs to make that happen. We also need to be looking at drug research. Developing new drugs, which isn't very profitable, but developing new antimicrobial drugs that work in a shorter period of time and a higher dose, and of course have fewer side effects. Again, our goal is to kill the bacteria and not give them enough time to figure out ways around the organisms. Now, our long-term strategy is trying to reduce the abuse of antibiotics, making sure antibiotics aren't given where it's not necessary. Making sure we're not giving antibiotics to animals. Animals are given so many different antibiotics. It increases their size. They grow faster. Problem is, that means we're ingesting those antibiotics, which is giving our bacteria more opportunity to develop resistance. And we should be trying to develop and take vaccines whenever possible to provide an alternative protection, so that we don't have to be taking those antibiotics and encouraging resistance. Now, other than the fact, we need to know which drug to take, and we need to know which dosage, and we need to take it correctly so that we don't have drug resistance, we do also need to consider, are there going to be any side effects? So about 5% of all people taking some type of antimicrobial will have some type of adverse reaction. to that antibiotic. And I'm like, and then, yes, sometimes I'm like, some of you might have some type of allergic reaction to some type of antibiotic, but you know it, and so you can avoid taking those certain antimicrobial drugs. But we need to know, is it going to have some type of adverse side effect to tissue? Are you going to have an allergic reaction? Another big issue, is it going to disrupt your normal flora, which can cause something known as a superinfection. Again, you have lots of normal bacteria that do great things for you. But if you take a broad spectrum antibiotic to kill a pathogen, it's not going to be the only thing that it kills. Just an example, think of the last time that you or someone you know has gone in for maybe an ear infection or a sinus infection. Has the doctor ID'd out what bacteria is causing that infection? Nope. They just, ask what are your symptoms, or look in your ears and say you have an infection, and they prescribe you a broad spectrum antibiotic. Well, its job is to kill the pathogen. However, because it's broad spectrum, it can kill a lot of your normal flora. And because normal flora do good things, other bacteria that are kept in check by those normal flora can start to flourish and cause side effects, bad side effects. This is just showing an example, and I'm like, that yes, you have lots of different organisms in your intestine, so many bacteria in your intestine. However, I'm like, when you take a broad spectrum antibiotic, again, we start killing the good bacteria in there. And these normal good bacteria are keeping these guys in check, because these have a potential pathogenic effect, which means they could become a pathogen. They could cause a disease, but they're kept in check by your normal microbiota. But if you take a broad spectrum and you kill all your normal bacteria, nothing's keeping these guys in check, and they're going to start to grow out of control. This is an example, or to show you an example of how this happens. C. diff infections is a superinfection. Your normal intestinal bacteria keeps C. diff in check. Everyone thinks, oh, I don't want to get C. diff. Well, you don't want C. diff, but a lot of people have C. diff as part of their normal flora. It's kept in check by your other normal flora. However, when someone goes to the doctor and they're given a broad spectrum antibiotic and they kill all their good bacteria, C. diff kind of has itself a little party, and it starts to reproduce like crazy because all the normal flora bacteria that has kept it in check for years, you've killed them all. It's why they try to encourage, when you are taking a broad spectrum antibiotic, eat yogurt. There's good bacteria in there. Take some type of probiotic, things to encourage the growth of your normal healthy bacteria so that you don't have super infections take place. Now, when we are trying to choose an antimicrobial drug, it's not just knowing, we have to know which drug to take, but we also have to know what dosage should be given. So we want to make sure we choose the correct drug. We don't want to send a patient home with an antibiotic and they take it for 10 days and it does absolutely nothing because it wasn't correct for the particular microbe they had. So we want to figure out which drug to be given and what dosage. So there are some tests that can be done in a lab to figure this out. This isn't always done right away. A lot of times they give a broad spectrum antibiotic, and if it doesn't work, then they're going to start to look for some more specific drugs for a particular patient. I'm like, but some of the tests they can be done, which are going to be in some of my next slides coming up, the Kirby-Bauer test, an E-test, and a test called the tube dilution test. But we do have to know when we're trying to figure out which drug, we want to make sure we know which drug works, but what is the nature of the organism that's causing the infection? What do we know about it? Where is it growing? Do we know what kind of genus it is? And what's the overall medical condition of the patient? Because there are some drugs that work together with a patient's immune system, that the two together, the antibiotic plus the patient's immune system, will get rid of the bacteria. However, if a patient has a weak immune system, you can't count on their immune system to help the drug. And so that might change which drug is given. Now, one of the tests to determine which antibiotic is going to work best is called the Kirby-Bauer disc diffusion test. Now, how it's done is, and in one of your labs coming up, your virtual labs, you'll talk about it, is they put bacteria all over a plate. So the bacteria is happily growing and give it everything it possibly needs. However, on that plate, we also put on top of the media little tiny paper antibiotic soaked discs. Different discs, sometimes different concentrations. Now, this one's showing different antibiotics. Each one is for a different type of antibiotic. And we're going to let the bacteria grow on the plate with those antibiotic discs sitting on the top. Now, we'll come back after 24 hours and look to see where is there the greatest, I'm going to say, effect of the antibiotic, shown by how far around that antibiotic are you not going to have any bacteria growing. So you get out a ruler and you measure this diameter, and the biggest diameter is going to be the most effective antibiotic. It's going to inhibit bacteria like way, because it starts to diffuse out and gets less and less, but it's going to inhibit bacteria way out to here. Whereas a drug like this, those bacteria grow almost right up to it. I mean, those bacteria are almost growing right up to the antibiotic and laughing in its face, where they're definitely not growing anywhere around this particular antibiotic. So we can test a whole bunch of different antibiotics against a particular bacteria that a patient has. So we can grow up the bacteria a patient has, put it on this plate, and grow it up along these different antibiotics to see which antibiotic seems to have the greatest effect against that bacteria. This is just showing how that works. Another is not only knowing which antibiotic works, but also knowing what dosage should be given. It's called the E-test. It's the effectiveness test. So these are kind of paper-soaked discs, but they have an antibiotic soaked into them at various concentrations. And we want to know, what's the minimum dosage of antibiotic? What's the minimum amount of antibiotic we can give to a patient that it's still going to inhibit growth, and it's going to be right at this bottom that it crossed? Because why give them a dosage of 32 when you can give them a dosage of 1 or 1.5 to inhibit bacterial growth? We don't want to overdose. One, it could cause more serious side effects, or two, the bacteria might now have to try to figure out a way around that strong dosage. Another is a tube dilution test. Again, looking at different dosage. This is looking at, we've added this different amount of dosage of antibiotic. And we want to know, anywhere it's clear, no bacteria are growing. Anywhere it's cloudy or turbid, bacteria are growing. I'm like, what is the dosage? And we know it's going to be right in here. What's the lowest amount we can give that we can stop bacteria from growing? Now, it's called the minimal inhibitory concentration, the MIC. It's just what's the smallest amount we can give that's going to inhibit microbial growth. Now, we have to compare that, and we have to what's known as the therapeutic index. What is the doses of drug that is still going to be toxic to humans compared to its minimal effectiveness, against us? What's the smallest amount that we can give to a patient that's still going to work, but yet have the smallest amount of side effects? So we want to know, what is the dose of drug that's toxic, to the organism compared to what its toxicity is to humans. And again, we want it to be more toxic to the humans than toxic to us. But again, the drugs aren't perfect. So everything in this PowerPoint, again, is different ways, different kinds of drugs, how they work, how they're developing resistance against all of the drugs that we're giving, how we can stop resistance from happening, and how we can figure out what drug and what dose to give patients.